Hello students, here is your bell work for today. Go ahead and pause your screens to complete the bell work for today. We're going to continue our talk of protein receptor types today and complete the lecture. So our receptor type we're talking about today is G protein coupled receptors, also called GPCRs. G proteins are transmembrane receptors, which means their protein touches both the extracellular environment and the intracellular environment, which is outside of the cell and inside the cell. This is the single largest category of receptor types in animal cells. G protein coupled receptors are going to sense molecules on the outside of the cell. So these molecules are the ligand, the chemical messenger that has a target cell in order to transmit this message. Hormones, light-sensitive compounds, neurotransmitters, pheromones, and even odors are examples of these G protein coupled receptors that use this type of receptor for communication. So what happens is the outside of the cell is going to start a signal transduction pathway within the intracellular environment that ultimately leads to a response from the target cell. They can either open a gated ion channel or cause an enzymatic reaction due to the G-coupled proteins' transformational changes within the intracellular environment. Here is a picture of an example of G-coupled protein. So you have this purple G-coupled protein with an alpha, beta, and gamma subunit. After the ligand is bound to the receptor complex, the G-coupled protein is able to shift right onto the plasma membrane where an effector protein is affected by this shift on the G protein alpha subunit. After we have this new membrane effector protein complex, that membrane effector protein complex with the G-coupled protein alpha subunit is able to further move to the right and causes a change opening this ion channel, which allows for the influx of ions into the intracellular space. Here is an animation that kind of shows how G-coupled proteins work. So you have your G-coupled protein. After activation, the alpha subunit moves over to this adenylate cyclase enzyme. ATP is then added to this other protein channel, and it stimulates the signal transduction pathway with the secondary messenger of C-AMP. And we're not too worried about C-AMP right now, but we are going to talk about this further on the subsequent slides. Let's watch it again. I'm going to let it start over. So we have our primary ligand, which comes and is about to bind to the red protein channel, allowing for the alpha subunit of the G-coupled protein to shift to the right and bind to the adenylate cyclase, which is stimulated also by ATP. Phosphorylation occurs, and the secondary messenger C-AMP is released into the cell to produce the rest of the signal transduction pathway. Here's a non-animated version of this. So we have the ligand again binding to the G protein coupled receptor. And the second, so it's called coupled because it has this G coupled protein receptor, the squiggly one, which is the transmembrane protein with the receptor on the external cellular environment. And the second part of the couple is the G protein. It is inactivated until the ligand is bound to the G coupled protein receptor. After the G protein coupled receptor is bound to the ligand, we have an active form of our G coupled protein where the alpha subunit is able to shift to the right and GTP is used and becomes GDP, which activates the G coupled protein. Now the alpha subunit combines with the GTP and separates from the beta and gamma subunits, binds to the effector protein and elicits some type of cellular response, and a resulting phosphate group is added into the cell. So this is smell you later because this is the transduction pathway of a bitter smell, trivalent salt quinine sensitive potassium channel. is the specific receptor that is responsible for the bitter smells that we smell. So after the ligand, the chemical messenger, which in this sense is that bitter smell, binds to the ligand, it causes changes within the intracellular environment. So after the ligand binds, we have the G-coupled protein activation, and this G-coupled protein activation is going to have the G-coupled protein subunits separate. We have phospholipase C becoming IP3, which means phosphorylation has occurred. And there's a secondary signal transduction pathway where we use C-AMP. That part's not important. All we're worried about is how the mechanism of the G-coupled protein receptor works. Please review these and understand mostly that G-coupled proteins can stimulate secondary messengers from their signal transduction pathway. Often the effector proteins activated by G proteins produce a second messenger, as I just stated. Two common effectors are adenylyl cyclase and phospholipase C. Adenylyl cyclase is the producer of C-AMP, and this is helpful for hormones that cannot get through the cell membrane but still want to elicit that cellular response. Phospholipase C, also called PIP, it's acted on by effector protein phospholipase C. It produces all of these different things, and both of these act as secondary messengers. And again, here is where we show the secondary messenger of C-AMP being activated by the subsequent ATP that is produced from the G-coupled protein shifting to the right, binding to adenylyl cyclase, and eliciting a cellular response. Different receptors can produce the same second messengers. So even though we have different receptors, many of them might still use the C-AMP pathway or the other secondary messenger as phospholipase C. Hormones, glucagon and epinephrine, in fact, can both stimulate liver cells to mobilize glucose. Different signals, but they both have the same effect. Both act by the same signal transduction pathway. So just because we have different signals doesn't mean we're aiming to target a different response from the same cell. Single signaling molecules can have different effects in different cells. Existence of multiple forms of the same receptor, subtypes or isoforms. Receptor for epinephrine, for example, has 9 different isoforms. It's encoded by different genes, and the sequences are different, but differ in their cytoplasmic domains. Different isoforms activate different G proteins, leading to different signal transduction pathways. Please pause and stay tuned for a description of your upcoming assignment and the exit ticket. All right, students, so here is the description of the Body Mechanisms Group project. Please view this and read all of the instructions. There are certain benchmarks that you guys all need to meet as a group. This project is worth 125 points, and if you work diligently and are all working together, and I see that your guys' work is very good, we will not have an exam for this unit. I repeat, if you work diligently, you meet your benchmarks, and everyone's participating effectively, then we will not have an exam for this unit. Please read the following benchmarks where there are certain things that you need to consider before submitting each benchmark. And consider this, that the leader will submit every benchmark as a PDF to the assignment, including all team members' names and the benchmark number that you are submitting for. Please get the benchmark number within the title so it's easier to view. Finally, after you guys complete your project, you will be required to submit peer reviews. Each of you will individually have to submit benchmark number 4 to this assignment so that you can best be graded on your work. The first benchmark, as you will notice, is due next Monday, which is about 5 days away from today when you guys are seeing this video. So please look at your groups start a Google document so that you can effectively work as a team. If you guys are still confused on how to see your teams, I will show you that right now. So what you're going to do is you're going to go over on Canvas to People, and you should be able to see who's in your group. The first thing I would do is start a way where you guys can all communicate with each other so that you are all able to communicate what needs to be done for the project. Within each of these groups, you will be able to have a discussion within your group by going to the groups page, and there you can discuss things, submit materials back and forth to each other, and discuss what needs to be done to meet the next benchmark. And you can also see who the student leader is within each of these groups. Whoever has this little person by them was the first person to sign up for that, or was the first person that I signed up for that group and is therefore the group leader. Please make sure that you ask me if you have any questions about this assignment, and I am happy to do drop-in sessions for groups that want to meet and make sure that they're on the right path to completing this assignment correctly. All right, please stay tuned for the exit ticket shortly to come from the lecture today. All right, everyone, here is your exit ticket for the day. Please go ahead and pause your screens to complete this exit ticket. Reach out with any questions. Have a great rest of your day.